Next up, we've got Dr. Ben Pascoe talking about Acinetobacter.
Thanks, Will. So my name is Ben Pascoe, and I'm a senior research fellow here at Oxford, and I've got a split position between the Ineos and the Big Data Institute. But today I've come to fill in for Evangelos Morkas, who was a postdoc with the group, and he's moved on to take up his own position in Sweden. I'm going to talk a little bit about how carbapenem resistance evolves in a contemporary collection of Acinetobacter baumannii isolates. So we've already seen this morning some support for how important Acinetobacter is. We saw a nice but not nice slide from Alina this morning, which had the number of deaths attributed to different organisms. And Acinetobacter was very high on that list. And as a result of that, the WHO has classified Acinetobacter as an organism for which is a critical priority to develop new antibiotics. And then also in America, the CDC classifies Acinetobacter as an urgent threat. So it's an important infection in human clinical infections, and multi-drug resistance, and particularly carbapenem resistance, really influences treatment options and how well patients recover from these types of infections. And in our story today, we worked with researchers in Russia, so Mikhail Edelstein's group in Smolensk, and he's collected Acinetobacter isolates from different hospitals and GP clinics from around Russia over the last 16 years, mainly in 3 different windows of collection rather than a traditional surveillance approach. But we used these isolates and these genomes to investigate carbapenem resistance. And you can see that these isolates come from various different infection types and locations, whether it's urinary tract infections or respiratory bacteremia, but then also different ward types, ICUs, and then outpatient clinics. We see a mixture of different isolates. In our collection, roughly twice as many came from male patients that came from female patients, and those that did come from female patients were from slightly older patients, and there was a slight trend towards isolates from older patients being resistant to a greater number of different antibiotic classes. So Mikhail's team as well, they looked at these susceptibility of these isolates, and you can see that over time we see lots of resistance to lots of different antibiotic classes, and in many of the different classes we see that this a steady rate of resistance, but in carbapenem, these 2 different purple plots, we see that originally in our first collection, not many of the isolates were resistant, but then in the more recent collection event, we see that nearly all of them were resistant. And this is consistent with a global picture. So on the right, we've got all of the genomes from the SRA and how many we predict to be resistant and how many we predict to be susceptible. And you can see that over the last 5 years or so, the rates of resistance have just shot up in this collection. So we wanted to understand a bit more about how does carbapenem resistance evolve? How is this? Are they all the same? Is it the same genes? Are there different mechanisms of resistance? And which mechanisms are most common in our collection? So we did this genome-wide association study. So this is, for those that are not familiar, it's a statistical approach where you have 2 different groups, whether that's resistant, susceptible, and we break the genome up, whether that's genes, SNPs, KMAs, or unitigs, and we use different methods to look for associations with our phenotype, so in this case, carbapenem resistance. So in these plots, each dot is a unitig. On the left, on the y-axis, we see a p-value or a probability that we think this unitig is associated with a phenotype. And the typical outcome of this is this sort of Manhattan plot, where we see these skyscrapers for hotspots in the genome that we think are important in this phenotype. And we did this for our 2 different antibiotics, these 2 carbapenems, and looked for overlap. And the ones that were the most associated with the phenotype were from these known genes, so they'd already been shown to be important in carbapenem resistance, and for those who are familiar, this BLA-OXA72 is a very common gene that will be acquired to give you resistance to carbapenem, but then also these linked genes or SNPs that might help you stabilize that resistance and keep hold of that resistance gene. And yeah, so when we look into the literature, you see evidence of this that supports our GWAS study, and this acts as kind of a positive control and gives us confidence in some of these results. And when we map these hits, these genes, back onto our isolate collection, so we've got a tree down the left of our different isolates, and these blocks are presence and absence of different genes and SNPs. And the first thing that you can see is it's quite blocky. There's areas of the population that all have the genes or all don't have the genes. So we see a sort of lineage acquisition of these genes, this resistance phenotype. But then we also see some relationships within this. So we see this BLA-OXA72 gene, which is in this grey area, which is a known plasmid. So we see a plasmid being acquired with a resistance gene, but then a SNP in the core genome that helps stabilize acquisition of that plasmid so that it can keep that resistance and express the resistance phenotype. And then we see a similar relationship in another part of the genome where we get a different BLA-OXA gene which is stabilized or which is moved around with transposons in this part of the tree. And if we zoom in on some of our isolates, we see this cluster of isolates. All these red isolates are Russian isolates. This white one at the top is the nearest isolate from another country. So we're seeing like an outbreak type tree, and we can see lots of red blocks where there's resistance to lots of different antibiotics. They all have this BLA-OXA72 gene with these linked SNPs to stabilize the plasmid. And if we include the time element, we can see that this sort of emerged in around 2010 and has almost emerged completely resistant to all the antibiotics that it was going to be resistant to. And we can use this sort of dating approach to support some of the epidemiology that Mikhail and his team have seen. So they'd also observed this cluster of isolates originally in Belarus, and then they found these isolates around the rest of the country. And they're asking, well, is that true? Is that the correct scenario? And we can use this dating approach to date isolates back to when the lineage emerged, although in this we've got a large error bar, so we'd have less confidence in the emergence of the lineage, but then more confidence in this recent time frame where we see lots of spread, lots of small error bars, where we're seeing spread of this lineage around Russia. So that's the kind of thing that we would like to do next, is overlay this temporal data, these genomic data, combine it with the geospatial data that we have from where the isolates were collected to support the epidemiology that Mikhail and his team are doing. And we'd also like to understand these relationships between mechanisms. So we've identified 2 mechanisms of carbapenem resistance. Maybe there's others, maybe there's other elements that are playing a role in these different mechanisms. And just a quick thanks to the rest of the team. So Evangelos has done all of this work and has led the project for the last few years. Sam Shepherd's group here in Oxford, as well as Kirsty and Tim, who have played an important part in getting the project going, and Mikhail in Russia, who's helped collect the samples. Thanks.